Patient with us. So now, ladies and gentlemen, I would like to invite with a big round of applause our honorable panel members. In the order of sequence, can I have with a big round of applause Mr. Amaresh Chakrasali, Corporate Quality Manager from Coca-Cola. Amaresh ji, welcome to the 3rd Annual India Food Nutrition Summit. Please come in, sir. Welcome. Joining him, we have Mr. Atanu Haldar, Senior Director, Herbalife Nutrition. A big round of applause to Mr. Haldar. Congratulations and welcome to the India Food Nutrition Summit. Welcome, sir. Joining them, we have Ms. Ranjeet Kaur, Lead Scientific Regulatory from Wrigley. Ms. Kaur, welcome to the India Food Nutrition Summit. Welcome, ma'am. Joining them, we have Mr. RATNESH SRIVASTAVA, General Manager, Research and Development, Foods and Beverages from Nutrelis, Agro Foods, our honorable moderator. RATNESH ji, welcome to the India Food Nutrition Summit. And joining them, we have Dr. Kansal, Technical Director from Kansal Agro. A big round of applause to Dr. Kansal. Kansal Saab, always an honor to have you, sir. Please come in, sir. So, RATNESH, all over to you. And a fantastic panel of expertise here. And food testing is a very important subject in the overall scheme of things for food nutrition. So, Captain of the ship, RATNESH, I give the panel to you and looking forward to a great session.
Myself, RATNESH SRIVASTAVA, I had worked with the industry in 25 plus years, the Dabur and Patanjali, and now connect with the Nutrelis Agro Food and also with associate with Geon Life Science. Now, welcome to Atanu ji, Herbalife, and Amaresh from Coca-Cola and Kansal ji from Kansal Agro, and Mrs. Ranjeet Kaur from Wrigley. Welcome to our team panelist. First off, introduce to what is the food. The food and any nutritional substance that people or animal eat or drink or absorb in the order to maintain the life and growth. First, I want to say, when we produce the food, first of all, there's the lot of the things, but 3-4 are major critical things. One is the how you produce the food. First is how to resource the raw materials. How can you connect with the raw materials? And another one is the how you processing it. And third one is the how you absorb and how you packed it and how you will deliver it. There's a 4 major content because the maybe food may be very tasty, good in the texture, good in the vitamins and minerals. But the more and more important, it should be hygiene. If it is not hygiene, that means high protein, high vitamin, high taste, high texture, there's no way. So first of all, the hygiene point of view is very important for the food in category, in overall. So when produce the product, we can go with the raw material resourcing, how you can resource, where you can resource, कैसे आप उसको लाते हैं, कैसे बनाते हैं. This is the very important. And how you can the process it. If you adopt the process, but you cannot pack in the proper structures of the laminate or something like that packing structure. This is also important. And if you have packed in the proper, but you cannot deliver it in the proper way, just like a milk, the frozen content and other conditions is adopt. This is very much important. If you follow all these things, your raw material is okay, your process is okay, but your delivery is not at the conditions. You cannot follow the processed food. This is the will not good the hygiene condition. And another was how you consumer adopt this. When you open the consumer once open the pack, and then how can you can define how can freeze out or not freeze out or what in the condition. Just example, you can open milk and you can purchase from milk farm market, and you just keep it in the side, but you are not keep in the fridge. Your product is the spoilage. Set like that. So there's the micro contamination is the very much important. Now our panel questions are there. The recent testing technology for R&D, rapid identification and application, importance of micro testing in food and beverages, need method and technology in the character of food and beverages. And another one, the food as assessment method using imaging technology. Now just ask to the question to my first member, Ms. Ranjeet ma'am. Just ask you one question. Mr. Ranjeet ma'am, how can we explain about the micro testing? How are the importance in the present scenario? You can please explain.
Thank you. So I think every one of us has experienced this COVID era. So I think we all understand what can an impact microbe can bring into our life. And as RATNESH has mentioned about the hygiene, I think we don't need somebody to tell us that what we need to follow and what we need to, test. I think that's where sometimes even the self-compliance piece comes into play. So talking from maybe a little bit from a regulatory aspect, that we know that the Food Safety Standard Authority of India has laid down certain guidelines with regard to the microbial sampling size, which you have to do for the various categories of products for which you have to do the microbial testing. And since we are all in the food business, so we know that there is specifically a microbe lab usually associated with our plants, because that's the main thing, that maybe you can compromise a little bit on the parameters, which may not impact safety. But microbial contamination is something which impacts majorly the food safety. And all of us, can get impacted by the recall which may be associated if it is found that our particular product is actually infested with microbial. And we also know that FSSAI has laid down the recall guidelines, and it talks specifically about unsafe food, right? And if some microbial has been found in the food, then definitely it turns around to be like an unsafe food. So apart from considering the various regulations, guidelines, which actually tells you that you need to do this, I think self-compliance is something which needs to be followed and imbibed into our operations, where we are working in. So I would say that always, take that at a priority, because the impact which it can have on the business can be huge. And it is an ever-evolving field, right? The method which you are using today, or maybe the technology which is available today, will upgrade itself. And sometimes there is a challenge that the technology needs to be actually aggregated as per to that lab. And there is always, a gap in terms of how a researcher develops a test, maybe like we talk about this rapid test, and how a regulatory body endorses the results coming out from that particular test. So we always have to fill up that particular gap whenever something comes in. But maybe just to conclude that, microbial testing definitely is in great importance in the food business. And we don't need somebody to tell us the guidelines. We need to be very much self-compliant on this particular aspect, because it can have an impact, hugely. So maybe just to tell everyone, I think all of us also know about the recall which happened in the Peanut Council of America, right? Which impacted, of course, the life of 9 people. And recall is something which actually lives in our mind for long, and it brings a lot of negative to the company which it is associated with. So I would say always build up those measures, whether it is with the prevalent methods, whether you make a rapid method, get the endorsement, follow what is being laid down in the guidelines, and definitely, you can have a smooth running business on that. So thank you.
Thank you, Ranjeet. And another members, Mr. Amaresh ji. Can you explain the rapid testing technology and detection for the application in food and beverages? Can you please explain?
Yeah, thank you. I hope I'm audible. Specifically coming to the rapid testing in food analysis and testing. If you see, generally testing, if you see as this one, that is the main factor in the entire, this one, which boosts the confidence. Okay, it it helps us to understand that whatever we are doing as a process, as a product, or it is complying to regulatory this one, testing plays an important role, as we all know. And when it comes to specifically the rapid testing, okay, rapid testing mainly evolved with regard to the microbiological testing or a specific these allergen testing or various things. So one of the things that was mentioning is regarding how real it is. If you see broadly, the rapid testing is based on either immunological testing or nucleic acid based testing, or like, PCR, RT-PCR. COVID has actually made it, RT-PCR, a very familiar household name now. Okay, and third, also biosensors are also developing very fast, and they also play an important role. So coming to the role of rapid testing in food industry, playing a huge role and important role. So when it comes to any testing method, reliability, reproducibility, accuracy, and the precision a test method or a testing process gives is very much important. So when we see a conventional microbiological testing method, yeah, they're all gold standards. They have evolved over a period of time, and then they're very dependable. They can see. But one thing which actually limits us in the current era, when the commerce is growing, when the business is growing a lot, okay, when there is, everything is fast. So rapid plays an important role. If you take a test which used to be take sometimes 8 days or 14 days, okay, with the commercial incubation and then in the regular incubation and then testing, it should take 14 days. Now with rapid technologies like flow cytometry, it is possible to do it with within maybe 2.5 days. That is the value it is adding. Along with all the benefits of the conventional testing, rapid is also adding turnaround time, productivity as a great commercial advantage for the industry. And at the same time, it also reduces certain things like laborious work, or maybe all that even the human intervention, human errors, it is reducing. So productivity is increasing in terms of handling a lot of products. So these are the things which actually rapid testing is catching up. And in the current scenario, if you see over a period of last 15-20 years, the methods are also developing. More and more pathogens are being methods are coming for more and more pathogens. So and more organisms. So that is where the trend is heading to, and also regulatory, while it is coming and developing, regulatory approvals are also coming in their own way in specific areas and pathogens. So that also would be a great developing thing in this. So this is the general and overall scenario where rapid testing is helping for us. And certainly I see a great growth as well as need, rather than the growth, need that is there for rapid testing methods for the industry.
Thank you, Amaresh. And another one is the participant is Mr. Atanu. Just asking one question. How these different rapid testing techniques can help for the quality control and assurance program of different
food industries?
Different food industry.
Okay. Thank you for the question. I think this is a very important question. As Amaresh was trying to articulate that how critical it is to have the rapid techniques and the evaluation method to be adopted by the industries and as well as the regulatory bodies. I think how this can be used overall at large by different industries is also going to be important. So I think the rapid rapid detection technique is not very new. I think it goes back, I think, a few decades. What I would like to say here is that the rapid testing technology can be used at the inception level, means when you are getting your raw material, right? And also in your process, like how do you maintain your hygiene and quality stuff, your changeover, like line changeover, so that, and also how this can be used in finished products. I think different industry will definitely evolve their own ways of how to use the rapid technology. Just for an example, at the raw material level, let's say for the dairy companies. Milk is such a company that we know, milk is such an ingredient. We all know that it is adulterated with many things, right? So, and National Dairy Development Board has come up long back with a kind of testing kit which can detect the adulterants then and there during the time of purchase of the milk from the farmers. Now for those industries who are using let's say different types of proteins, dairy protein, plant protein, we know that the dairy protein has got certain amino acids which are predominant. Let's say for, take a name like isoleucine for dairy protein. For, let's say, plant protein, it's a methionine, right? So how can you quickly test that, yes, the raw material that I am getting is actually a soy ingredient, or whether it is a dairy ingredient, right? So you can go in for some kind of, let's say, NMR technology, and you can reference it with your reference, and then you can tell convincingly that this is my dairy protein, this is my soy protein, or any other plant protein to that extent. Then there are methods like aflatoxin, which mostly used by which is a cause of concern for many industries who are into cereals and pulses, right? And we have the techniques, rapid techniques to take to detect presence of aflatoxin, like aflatoxin M in case of dairy is also very important, Then there are certain raw materials like let's say preservative or some functional ingredient which has got certain functional group. Right, so you can also detect the presence of those functional group very quickly, and you can ascertain, yes, this is, let's say, sorbic acid, or this is my some other preservative, right? So based on the functional test also you can do, and quickly you can use the techniques like, let's say, NIR technology, right, near infrared technology for such purposes. Then there are some, let's say, herbal product. You can immediately check for the presence of the active ingredient, which could be done by either by your NMR method or by NIR method, and then you can tell, yes, my raw material is correct raw material. Okay, let's say if you have the PCR, which Amaresh was talking about, if you are having probiotics, whether you have received the right strain or not, you can check using the rapid detection system. Now coming on to, so these are about the inception level. Now coming on to your plant quality hygiene, and then equipment hygiene, your contact surface hygiene. I think we all know that this particular field, the microbiological rapid detection system, has evolved in last 1 decade or so. I mean, exponentially, I would say. And even just now I had been talking outside with Merck people, and get to know that you can test like anything, the total plate count, the yeast-and mold, the E. coli, and then the salmonella also, right? They were much quicker. I think it's yeast and mold. We cannot imagine that it can be tested in 2 days, and so these are going to be really helpful. When I'm cleaning my, let's say, CIP cleaning, right? CIP cleaning, I can do some kind of, test immediately for the presence of protein or, let's say, any gene, right? So these are going to be immensely helpful. In Herbalife also, we have been using such, methods for rapid detection. What we use is basically that's ATP, what is that called? Bioluminescence. So that gives you the result in seconds, and you are confident that, yes, my line is now hygienic, I can start my production, right? So at the production level, you can use like this. At the finished product level, sometimes what happens, before you make your finished product, let's say, take an hypothetical example of some nutrient powder, which is spray dried, right? Before doing the spray drying, you go for the blend check, right? So the blend check, you pick up some markers like this vitamin or that, let's say, mineral you want to test. You can do those tests very quickly, and then you can feed that into your spray dryer, right? So that saves time, that gives you more confidence that, yes, the product after I spray dye, it will comply to my specification, right? And also at finished product level, there are techniques like, although these are not very accurate, I would say, but can give you certain direction that whether your approximate composition of the finished product is correct or something you need to do. So I think there is a huge scope that how the rapid detection technology can be used by the industry at large.
Just to add on, it's not only the commercial part or productivity. Rapid testing also helps a big way in pathogen breakout prevention and all that. It helps us for quick decision making, whether to release a particular product in general. So where it used to take 5 days, maybe the decision can be taken in 2 days. So pathogen outbreak containment is also one of the important thing that rapid techniques do help us.
I think as he was talking, I think one thing came to my mind is that yes, the regulators, as a part of their surveillance program, I think they can also pick up samples and go for certain testing, right? Which the regulators feel that the product is little susceptible, right? So I mean the adaptation of the rapid technique is going to be the success, I think, for the food industry going forward.
Thank you, Anatuji, and thank you, Amareshji. Thankful for this output. Mr. Kansalji, you please give me some inputs from your side.
That we have tried to introduce our new product. Earlier, there were, we introduced odourless egg, was the first odourless egg. That is the lot of enthusiasm was there that in India, in India, there are producing in the world, the first odourless egg that was released in 2009 and December at the very nanotechnology conference was there of the vice chancellors and NDRA canal, and the egg was introduced at that time. And that the egg should be free from all the type of material which is not useful to the human body. Mainly the pesticides are there, or the fungi are there, or the aflatoxins are there, or number of other products which are there, and the heavy metals which are there, or number of them. These all are the, as the rapid test, all the NAVL labs are there, where the tests are being done, and rapid testing that is done. And on that basis analysis, the vitamins, the hormones, whatsoever the things are there, we want to do, that can be done in the world, in the rapid testing method. And our feed and water is also tested, feed is tested, water is tested, even the droppings of the birds, which are in the case the ladybirds, that our farm is totally free from Salmonella, E. coli, other type of bacteria which are there, which are not there. And the feed is totally free, and the birds are also totally free. So we call it as the totally free, which can be used for the human consumption as a whole. And because odourless, which are mixing 16 type of herbal products in the feed of the birds. See, like the tulsi, amla, bheda, number of other products are there, which in neem or they are useful to the human body. They are useful to our culture, to things which are there. And similarly, we developed at that time this odourless. Now we have come to with the other product also, that is called, which is divit egg. That divit egg is that we have a lot of problem of divit patients, or the vitamin, say divit 2 type of problems are there, number of persons are there, which they are not able to consume. They are consuming they are not consuming the eggs because the eggs are rich in fatty acids, fats are there, and those fats are 2.5 times are more rich in, therefore, in carbohydrates, which lead to the more problem of the, say, more diabetes. But we have to produce that diabetes egg. This is a first international egg, which is there also, which I have been doing, that we have mixed three more, say, three things are there, but say, we call it a Vijayasar, Paneer Dodi, and Methi. Normally, Methi is given to the human beings. Maybe doctors are recommending the medicine, and it's a type of, say, that lowers your sugar level, lowers your in the animals, in all type of here. Total production is there, maybe in a different form there. Now the second thing is the, say, that is called as the Vijayasar. Vijayasar again is then Vijayasar is a product which is using protecting your pancreas. Pancreas in the body, that is the pancreatic cells which are secreting the insulin, the production of those things are there, that the insulin level, that your body, pancreas is protected, means the insulin secretion is there from the pancreas. That pancreas are to be protected. And at the same time, say, having a useful function for the pancreas, even the need is there, these wounds are there, number of other things are there, blood pressure there, the heart problems are there, then the pneumonia is helping, say, equalizing the blood pressure and the usefulness in the conversion of the, say, fats into a good cholesterol, good type of cholesterol instead of bad cholesterol. So mixing all these things together, protecting your body, at the same time, though in the, we see that the normal number of medicines are there. Number of medicines for controlling the sugar level. Say you can maintain the sugar level, but there is trouble that the sugar level can affect your body in number of ways. Maybe from the heart, maybe from the brain, from the number of organs in the kidney, or slowly and slowly, as the age group is increasing, the level of insulin production in the body is decreasing. There is a sort of increase in the, say, decreasing in there. There is a sort of weakness, trembling is there, conditions are not there. Normally the number of urination which you are going for number of times go for urination. Not one time.
Excuse me, Dr. Sir. Excuse me, Dr. Sir. Our content is the importance of micro-testing in food and beverages and need to method the technology. Please explain about this.
That is the main thing is our aim is the protection of the body.
Yeah.
Say that is the main, and then that is, you can have the sugar level maintained, but this help your reduce your say sleep is, sound sleep is there, say not go for urination, and 6 to sometimes have a good urination. In the winter sometimes I have to wake up for 3 or 4 times, go for the urination bar, again come to sleep and we are not sleep.
Yeah, Dr. Saab, we'll come back to the topic. In the meanwhile, we'll come back to that topic. In the meanwhile, maybe we can take the next one and then come back to him. If you can go ahead with that.
That is the main thing. The third is you have a exercise also, lot of.
So maybe I think we can just,
Yeah.
we'll come back to that topic.
That is the main thing is our aim is, I will be testing all these things, that our 3 things which I have mentioned to you are detected, say estimation is done in the egg. whatsoever the egg is there, the estimations are done. And the pH of the egg, that is 8.6.
Right. So maybe I think we
No,
come
I
back
think
to this
we-
and we'll do on this. So maybe we were discussing about the advantages of the rapid testing. So I think even though there are a lot of advantages of the rapid testing, but we need to control, like, what are the flip side of it, right? We like to have, good time that, okay, fine, we have technology, let's start to use it. But we should not forget about the methods which are existing, right? We should keep on, working around that how this particular new method which is being developed to have some scientific data points, at least, for a regulator to make that, okay, fine, this is something which can be used maybe for a wider population or a wider scope of it. So I think it should go hand in hand when you are developing, a new method. You have to also invest in terms of the research which you do for that particular method in order to generate some data points, so that at least, when a particular regulator is looking at that point for a mass consumption, then at least that is available. It should not be left that somebody made it and somebody starts to use it and then you leave it all, right? So it has to be like a complete chain or a cycle which has to be maintained and followed,
Yeah.
right?
The new product, just we have distributed this.
Yeah, that's an excellent point. That's excellent point actually. So for any rapid micro testing, definitely we discussed about advantages. But when it comes to disadvantages, yes, not exactly disadvantage. The further focus is that a good regulatory framework and then a proper verification and validation protocol need to be built in, so that the reliability and the correlation between the conventional gold standards always is made, and they, whatever results come either through rapid testing or conventional testing matches and goes hand in hand. That is one of the important thing that certainly we need to focus. And another disadvantage, if I want to say, every particular microorganisms or any specific test needs a long method development, testing, and a longer period to establish it both in the industry as well as in the market. Okay. Ratnesh, over to you.
Thank you for overall, everyone. Any question from? Anyone question from audience? Yes.
Yeah, someone from there.
Can you give the mic, please?
And your company name and the name of the person to whom it is addressed from the panel.
Yeah, my name is Anant Padmanabh, Quality Head for Big Basket, private label. My question is to Mr. Amaresh. What are biosensors he mentioned? And also in rapid testing, how do we avoid any interference?
Okay, I'll answer the second question first. Yes, in any testing methods, whether it is chemical or biological, and avoiding interference and background noises is an important subject. I'll just take an example. For example, in case of flow cytometry, okay, there can be the cells of interest which actually need to be detected and quantified. That is one. But the noise or interference mainly comes with things like, damaged cells or debris. There all comes. So basically avoiding the noise and interference has different techniques. One of the thing, like in case of flow cytometry, how it is done is, see, the flow cytometry depends keenly on the way light is scattered. Okay, and the detector detects it. So in that case, so there is a threshold that is set. The scattering pattern by the interfering debris or the damaged cell is different than that of a whole cell of a pathogen or any micro that is of interest. They differ. So there is a threshold that is put to differentiate between them, so that the differentiation is done, and the whatever is not reaching the threshold, that is kept away from the final results or the graph that you get out of this one. Okay, and on biosensors, see, biosensors mainly, what happens if you see the name bio and sensor, a biological activity or a physiological activity, the signals that produces, okay, it is basically a bio component and an electronic component. Okay, so it may be based on optical properties, it may be based on electrical properties, or it may be based on various things as voltage or piezoelectric, various things would be there. So the signals generated by biological activity, it may be an enzyme based or it may be any immunological based, so that is actually captured, whatever signal is generated is captured by the electronic sensor. Okay, so that particular signal is actually transmitted, analyzed by the software, and given in the form of an output what is required. This is a highly developing field. It's used both in the chemical analysis as well as in the microbiological analysis. So it's a highly developing field based on all these 4 techniques and combines both electronic as well as the biological component and activities. I hope that helps.
Any further questions from the audience? I think we have one more question.
Yeah, morning sir. This is Pawan here and I had a question on NIR technology.
Sir, can you kindly take the name of your organization as well?
Yeah, I am Pawan here from MTR Foods, Bangalore. So during our discussion, we had a topic on the NIR technology. So we talk a lot on the rapid testing technologies and incorporating those rapid testing technologies into our testing facilities. But there is also lack of validation results or the confidence to adapt those technologies into our day-to-day testings. So I feel that there should be some validation or some proper data built up to gain the confidence that whatever results we are getting, the reliability and the repeatability of the results what we talk. So how to bridge those gaps?
Any particular person from the panel to whom that is answered?
I would like to address this question to Mr. Amaresh sir.
Amaresh ji, I think.
See, whether it is FTIR, NIR or Raman spectra, any spectral analysis that is there, it might not give the same, this thing, but they are very reliable. Okay, I'm exactly not getting the point where the unreliability comes from. So the reliability factor of these testings certainly depends on how well the library is built, how well the validation is done, and how well the correlation is done. If these 3 things are there, so certainly there should not be any element of lack of reliability or confidence in that. And since they give quick results, they are of great value to the industry. So, but if you can exactly discuss about maybe after the session, like, why the element of unreliability comes. Is it because of lack of the historical data, or it is because of the hesitation to move to a new technology? So that can be discussed. But certainly building a good validation framework and validation method and correlation will give a lot of confidence. They are being widely used.
Thank you. Do we have any questions from the audience? That would be the last question. So we have one question. Kindly do mention your company and your name and the panel member to whom it is addressed.
Myself, Manish Gupta from Xeon Life Sciences. My question is to everyone, after the COVID, there is a change in the parameters of the testing of the food globally and also as per the Indian standards. How India is competing in that segment? How we are global standards are equal as per of Indians, or how is it changing now?
So I think Ratnesh ji, since it's a question directed towards the entire panel, maybe you can decide who from the panel member would be the right person to take it.
Okay, I would like to express my view first. See, all these methods, whether used in Europe or in US or in India, okay, they have similar protocols, they have a lot of uniformity. Okay, when it comes to a global comparison, India has a lot of capability, dependability, and also reliability in terms of testing. There is no dearth of any kind of you can say a lesser value or lesser dependability in India. The testing procedures go very well. If there are any intentional deviations done, that is different. But from a capability point of view and from dependability point of view, India definitely is at par. Okay.
I hope that answered the question. So ladies and gentlemen, I think a big round of applause to this wonderful panel here. And before we bid adieu to this beautiful panel for enlightening us on food testing, I would like to call on stage Mr. Avinash Woody, Director Product Marketing from Manage Artworks, to kindly felicitate this amazing panel here. Avinash, welcome to the stage of the 3rd Annual India Food Nutrition Summit. Thank you once again, honorable panel members, and I would ask Avinash if you can join them for a quick photograph. A big round of applause for this wonderful panel. Mr. Amaresh, Atanu ji, Ranjeet ma'am, RATNESH ji, and Dr. Kansal. Thank you so much, panel members, and thank you so much, Avinash, for the felicitation.